Hello, I'm Krishna Nukala, a graduate student in the Madhav lab in the Department of Biology, and today I'll be talking to you about how the different pathways of epilepsy, neurodegeneration, and the immune system are connected. So, to begin with, epilepsy is a neurological disorder that is characterized by seizures, where disruptions in the function of several genes are also known to contribute to the generation of seizures. With epilepsy affecting around 1% of the population, it is alarming to know that the currently available treatment methods are largely ineffective for a third of the epilepsy patients. So here in the Maddox lab, we study a gene called prickle, which is known to act as a compass of an individual cell. For example, prickle is known to help guide growing nerve cells to reach their targets, and they're also known to facilitate the transportation of cargo within the nerve cells. But when the function of the prickle gene is disrupted, specifically in the nervous system of a fruit fly, it is known to cause epileptic seizures, similar to those found in human patients with mutations in the prickle gene. So let me show you a video that has been slowed to 1/10 of the speed, showing a fly with a mutation in a prickle gene and is experiencing what is known as a myoclonic-like seizure. So, a myoclonic-like seizure is a seizure condition that is characterized by repetitive jerks of isolated muscle groups, such as the muscles in your limb. And here's an example of another fly that is experiencing these myoclonic-like seizures, but progresses to become a full-blown tonic-clonic-like seizure, something that we typically see in human patients with mutations in the prickle gene, therefore making these flies a great model to study the disease. And since we know that there is a lot of interplay between the nervous system and other major biological pathways in an organism, we wanted to know what other pathways are connected to the seizures occurring in prickle mutants. So for that purpose, we specifically looked at the brains of prickle mutants, and using computational and molecular genetic techniques, we found evidence of a heightened immune response occurring in the brains of prickle mutants. With recent studies pointing out the detrimental effects posed by a rogue immune system in the brain, we wanted to take a closer look at the brains of prickle mutants under a microscope. And similar to what we see in Alzheimer's patients, we also found evidence of a significant increase in nerve cell death occurring in the brains of prickle mutants as they aged. And by analyzing the behavior, we also found that the seizures in these mutants also got worse as they aged. So this led us to hypothesize that the dysfunctional nervous system in prickle mutants is contributing to a robust increase in brain immune response, which is leading to a significant increase in nerve cell death in the brains of prickle mutants, and in turn exacerbating the seizure condition in these flies as they aged. So remarkably, when we decided to suppress the immune system in the brain using powerful genetic toolkits available in the fly field, we were able to significantly reduce the amount of nerve cell death occurring in the brains of prickle mutants, as well as significantly suppress the exacerbation of seizures in these mutants, therefore showing for the first time that genetic suppression of the brain immune system can lead to the alleviation of the seizure condition in prickle mutants. Therefore, we believe that this novel connection between the brain immune system, the epileptic seizures, and the neurodegeneration can open up a new avenue of anti-seizure medications in the future. So with that, I would like to thank the members of the Manic Lab who have been working on this project, as well as our collaborators and the funding sources for the support. Thank you.